Morning, guys. We're going to talk today about Chapter 5. It's cellular reproduction, and we're going to talk about how mitosis and meiosis happens. So, um, remember that all multicellular organisms begin life as a single cell, and that cells can only come from previous cells. And reproduction continues as we grow to replace those worn out or damaged tissues. It's said that every 7 years you become a completely new human because you have replaced all of the cells in your body. You can see how cellular reproduction happens in the way that children grow, in the fact that we make scabs and scars. That's those tissues repairing themselves. And even in watching a fetus develop from a single cell all the way to a fully formed human. So in asexual reproduction, which is mitosis, that doesn't require the sperm or the egg, which is required in cellular reproduction. And there's two important processes that happen in mitosis. There's growth, where the cell duplicates its contents, including its DNA and its organelles. So if it had one mitochondria in that cell, now it's going to have two. And then cell division, where that one cell cuts itself in half and becomes two new cells. So it's heavily regulated by different enzymes and proteins and hormones. And so we're going to talk about that as we go. So chromosomes are DNA that is combined with proteins. That is what makes up the genetic code for when you're trying to make those new cells. It's a full set is passed to each daughter cell, and it allows for easier distribution to those daughter cells. Chromatin is that DNA and those associated proteins, and it's what we can see by using those high-powered microscopes. Not what we have in the lab, but what we can see. Usually with some electron microscopes, we can see it really well. We will be looking at some cells, not this week, but next week, that are going through that mitosis and meiosis process. So DNA is wound around something called a histone to form a nucleosome. And just before cell division, that chromatin condenses into chromosomes. You can see in this top picture where all of that DNA has coiled up into those two chromosomes that are hanging out right there. Humans have 46 chromosomes. Your sperm cells and your egg cells have 23 chromosomes, so that you get half of the DNA from your mom and half from your dad. And we'll talk more about that when we get to meiosis. So the duplicated chromosomes are composed of what they call sister chromatids, and they're joined at a centromere, which is this little center part right here. And each sister chromatid has identical DNA. We will talk about how sometimes there are crossovers that happen in meiosis, but that does not happen in mitosis. So the cell cycle goes through a bunch of phases, but there's three main parts that it goes through. That's interphase, mitosis, and then cytokinesis. Interphase is the regular, just hanging out being a cell. Mitosis is where it starts to pull apart that DNA and start to divide. And then cytokinesis is where that actual division happens, where it cuts itself in half, cleaves itself in half to become two cells. So this kind of shows you how the cycle works. This is a really good visual. So we're going to leave it up for a little bit while I explain kind of what's going on in each of these steps. This is the entire cell cycle. So this is interphase, the mitotic phase, and cytokinesis. So in this top G1 phase, this is going to be where growth occurs in that cell, so that when it cleaves in half, it's not going to be smaller than a regular cell. And it has to make a decision if it's going to divide or not. If it doesn't, it goes into this G0 phase, and then it'll come back to the G1 phase. But this is where it spends most of its time as its cell life happens. Then we go into this S phase, and that's where that DNA replication occurs as chromosomes duplicate. So that's where those sister chromatids start to be created. And then we go into this G2 phase, and that's some growth that occurs after the DNA replication. So again, it won't be a smaller cell than it needs to be. And then we go into, I don't know why it's doing that. Okay, then we go into mitosis, and it's actually split into some extra phases. So there's prophase, metaphase, anaphase, and telophase. And after all of those phases, then cytokinesis happens, which is where those cells split in half. So, like I said, the majority of the cell cycle is in that interphase, and it's when it performs its usual function. So if it's a liver cell, it's doing liver things. If it's a heart cell, it's doing heart things. And the amount of time that it spends in this interphase depends on what type of cell it is. Depends on if it's a liver cell or a heart cell or a muscle cell. And again, we said that it has those 3 stages. In that G1 stage, it's going to double its organelles. If it had 1 mitochondria to start with, then it's going to have 2. And it starts to accumulate some materials for that extra DNA synthesis. So starts to acquire more nucleic acids so that when it does need to make that sister chromatid that's exactly the same as the previous chromosome, then it has enough nucleic acids to do that. And then it, again, makes that decision whether to divide or not. In that S phase, DNA synthesis happens. So it makes a copy, essentially, of that chromosome so that it has those sister chromatids. So that when we start to go into that mitotic phase, when they split, both the cells will have identical DNA inside of them. And then that G2 stage follows the DNA synthesis, and it extends into that onset of the mitotic phase. And it synthesizes the proteins that it's going to need for cell division, because it doesn't just happen automatically. It's going to need some proteins to help split that plasma membrane. It's going to need some lipids to make a new bilayer, and that kind of stuff for when we make those 2 daughter cells. So the mitotic phase, cell division occurs. It's the division of the nucleus and then the division of the cytoplasm. So in the mitotic phase, the nuclear contents of that parent cell are going to be distributed into 2 daughter cells. So when it splits, that's what's called the daughter cell. Each of those sister chromatids has the same genetic information, and daughter chromosomes are separated sister chromatids. So we start out here with this one chromatid, and then chromosome. And when that S phase happens right here, then it produces these sister chromatids. And then when mitosis happens and they split apart, then they become daughter chromosomes. So the daughter chromosomes go inside the daughter nuclei that are produced by mitosis, and they're genetically identical. And every animal has an even number of chromosomes. Each parent contributes, like I said, half of those chromosomes. So when we were talking about species and how usually species can't breed together, it's because of that number of chromosomes. And like we said, where horses and donkeys can breed together, their offspring are sterile because they have an odd number of chromosomes. In the drawings that you might see of meiosis, colors are sometimes used, like a blue and a pink, to identify which chromosomes are contributed by the male and which are contributed by the female. But again, these are just artist renderings. So one very important part of mitosis is the spindles and the spindle fibers. So almost all eukaryotic cells rely on these spindle fibers to pull those chromatids apart. They are part of the cytoskeleton, and they're made up of microtubules. The centrosome, which is that part in the middle, is what those spindles attach to be able to pull those sister chromatids apart into daughter chromosomes. They may overlap at the spindle equator, which is the part in the middle, and they attach to duplicated chromosomes. So mitosis is a continuous process. It's happening in your body all the time, but it's traditionally divided into 4 phases: prophase, metaphase, anaphase, and telophase. So plant and animal cells differ in the way that they complete mitosis, and we're going to talk about that as we go through. So plants have centrosomes but lack centrioles. In animals, each centrosome has 2 centrioles and an aster, or an array of microtubules. And I know those words don't mean a lot to you, so let's look at a picture. So this is mitosis in the plant cell. In this inner phase, it's just out there being a plant cell. It's just living its plant cell life. And then it goes into prophase, where it's starting to condense that material that is made in the S phase into those sister chromatids. And then here you can see that those sister chromatids in metaphase are lining up along this equator here in the center, so that they can be grabbed by these spindle fibers and pulled apart. So this is the anaphase, where they are being pulled apart to the 2 poles of the cell, because remember, this is the equator. If we think of it like the Earth, this is the equator and these are the 2 poles, and so they're being pulled apart. And then in telophase, they're all the way pulled apart, and we're starting to cleave here in the center. So although it's divided into phases, it just happens continually. It's not like this is metaphase and it lasts for this long. It just continually happens. It almost happens imperceptibly, but we are able to take those quick snapshots so we can see those different phases. So since the DNA is replicated before mitosis begins, then those chromosomes start to bundle together in the center. They start to line up to be pulled apart. So mitosis is usually followed by cytokinesis, which is dividing of that cytoplasm. It begins during the telophase and continues after the daughter nuclei have formed. So here's an artist's rendering of an animal cell. And this is interphase, like I said, where it's just hanging out. It's replicating its nucleus. It's duplicating those organelles. And it's just living its best cell life. And then as we go into the cell cycle, the first phase of mitosis, that's prophase. And you can see that the chromosomes are starting to form, and the sister chromatids are forming up. And the centrosomes are starting to make those spindles. You can see this is an actual scanning electron microscope view of this happening. So they're starting to form up. So then in metaphase, the chromosomes get aligned right in the center. And the spindle fibers extend to the poles, where they're going to pull those apart. And you can see these are the chromosomes here in the equator, and they're going to be pulled apart. And so in anaphase, that's where they get pulled apart. So that's where those equal numbers start to pull apart into 2 sides. And then this is telophase and cytokinesis. So you can see that those chromosomes are in their own daughter nuclei, and they're starting to cleave apart. So cytokinesis in animal and plant cells, it accompanies mitosis in most, but not all cells. So it can result in a multinucleated cell, for example, muscle cells in vertebrate animals and embryo sacs in flowering plants. They have multinucleated cells. That means that they have more than one nucleus inside that cell. usually because the muscle cells have so many proteins that they need to make. They need a little bit of extra brain power, as it were. So when that cleavage furrow forms as anaphase ends, there forms a contractile ring, which is some of those proteins, those actin filaments, and they pull it like a drawstring, and those contractile rings, I don't know if you've ever banded a calf, but it's like when you're trying to put that band on the top of the testicles, and it just starts to cleave it off. So it just pinches it until it just falls off. So that's kind of what happens in these animal cells. Plant cells, remember, they have those cell walls, and so that prevents that cleavage furrow. So instead, it builds a new plasma membrane and cell wall between the daughter cells. So the Golgi body has to produce those vesicles that then take those lipids, the phospholipid bilayer, and starts building a new cell layer and then a new cell wall. So that's the basics of mitosis. Make sure that you know each of the phases, the interphase, the prophase, the metaphase, anaphase, telophase, and cytokinesis, and what happens in each of those phases. So now we're going to move on to meiosis. So this is where we're going to create sexual reproduction cells. It reduces the chromosome number, because if you had all of your chromosomes from your mom and all of your chromosomes from your dad, then you wouldn't be a human, because you would have 92 instead of 46 chromosomes. And then it shuffles up those chromosomes to produce some genetically different gametes. So it adds a little bit of genetic variation in both plants and animals. So we have something called a homologous chromosome, and those are members of a pair of chromosomes. They're also called homologs, and they have the same size, shape, and construction, including where the centromere is on them. And they contain the same genes for the same traits. Those are homologous chromosomes. So when they split apart, they're going to have the same information. So a child receives one member of each homologous pair from each parent. So they may contain different versions of the same gene, but they're going to contain the same gene. So alleles are alternative forms of a gene. So what that means is you might get a gene for brown eyes from your mom and blue eyes from your dad. They're the same gene for eye color, but they express in a different way. So males and females have 23 pairs of chromosomes. And so 23 pairs equals 46 total chromosomes. And that's a diploid number. Your gametes, so your eggs and your sperm, are what we call haploid. And so they only have 23 chromosomes. There's 22 pairs of autosomes, and those are basically everything that makes you, skin color, eye color, the way you put on fat, the size of your bones, the shape of your bones. And then you have one pair of sex chromosomes, and that's what determines whether you're male or female biologically. So here are those homologous chromosomes of a male. You can see that males have an XY chromosome for their sex chromosomes, and females have an XX. So you can see how they're the same size and shape. You can see how this, maybe this is the gene for eye color, and this is the brown gene that they got from their mom, and this is the blue one that they got from their dad. It looks a little bit different, but it's the same size, shape, and construction. So in sexually reproducing organisms, the life cycle refers to all the reproductive events that occur from one generation to the next. So it involves both mitosis and meiosis. So mitosis is the continued growth of the child and the repair of tissues throughout your life. And it results in those diploid numbers. Remember those 46 chromosomes. Meiosis refers to that creating of sperm and egg cells, and it reduces the chromosome number from diploid to haploid. So remember, diploid is 46 and haploid is 23 in humans. So it reduces it in half. Spermatogenesis is when sperm are produced in the testes, and oogenesis is when eggs are produced in the ovaries. Males continue to produce sperm throughout their life, but women are born with all of the eggs that they're going to have for their entire life already in their body, and they're just released through the menses cycle. When those eggs and sperm join back together, they form a diploid zygote. So it goes from diploid to haploid back to diploid. So you have your male and your female, and they produce sperm and egg cells through meiosis. And then when they join together, that's called fertilization. And then it forms that zygote, and then that splits by mitosis to make their baby. And then it continues to split through mitosis until it forms a fully formed woman or man, and then it goes back through that cycle. So where mitosis results in 2 daughter cells, meiosis is going to result in 4 daughter cells. The reason is because we're trying to reduce that number by half, remember? So before meiosis 1, each chromosome has duplicated, just like in mitosis, okay? So, and then it goes through 2 divisions. So meiosis 1, those homologous pairs line up, and they split in half, just like in mitosis. And then in meiosis 2, they don't duplicate those chromosomes again, but then they split them apart one more time so that they have 4 individual cells. So here's where your chromosomes duplicate. So meiosis 1 happens, and we split those into 2 daughter cells, just like this. And then meiosis 2 happens, and we split them into 4 daughter cells. Okay, so we have doubled the number of chromosomes here, just like we would in mitosis. If we were just doing mitosis, this split would result in those 2 daughter cells that have identical genes. But remember, we're going to go through one more split so that we can get those haploid numbers. So something that happens in meiosis that does not happen in mitosis is crossing over. So during prophase 1, those homologous chromosomes pair up and form a tetrad. And each tetrad consists of 2 chromosomes, with each chromosome containing 2 chromatids. So tetrad, like meaning 4, and there's 4 chromatids. So it looks like something like this. So when that happens, some of the genetic material from here can switch over to here. And let me show you what that looks like. So when a tetrad forms during that synapse, those chromatids can exchange genetic material, and that's where we get genetic variation. It increases variability of the gametes and therefore the offspring. So you can see here where we've got those non-sister chromatids. So remember, sister chromatids are the ones that are connected with the centromere, and then the non-sister chromatids are the ones that are paired up in this tetrad, and those 2 kind of cross over and exchange this genetic material. So then when you split those into 4, you've got some more genetic variation. They're not all the same. They still have the same genes, so they still code for eye color, but this one may code for brown, this one may code for blue, this one may code for green, and this one may code for like gray or hazel. So the reason that meiosis is so important is because we want those chromosome numbers to stay constant in each new generation. And it also generates these genetic variations. This is important so that, everyone doesn't look the same. And so that there's genetic variability. So maybe you carry a gene for sickle cell anemia and you breed with somebody who doesn't. Then your kids have a better chance than if somebody who had sickle cell anemia bred with somebody else who had sickle cell anemia. Then their kids would have a really good chance of getting that sickle cell anemia. So we're going to go through the phases of meiosis, just like we did with mitosis. It involves 2 divisions, and each division is broken down into those 4 phases. So in meiosis 1, there's prophase 1, metaphase 1, anaphase 1, and telophase 1. And then meiosis 2 has prophase 2, metaphase 2, anaphase 2, and telophase 2. So here is meiosis 1, and it happens just like mitosis, okay? Same thing happens. We produce those tetrads. The one difference is in this prophase 1 that crossing over happens. And then metaphase, we're going to align those chromosomes up. In anaphase, we're going to split them apart. And then in telophase 1, we're going to split into 2 cells. But that's not where it ends, because remember, we have to create 4 individual cells. So then we go to meiosis 2. And in prophase 2, the same thing happens. But this time, there's no crossing over. And we're going to split those sister chromatids apart so that we form 4 cells that are genetically different from each other and from the parent cell. So meiosis has 2, this is meiosis compared with mitosis. So meiosis has 2 consecutive nuclear divisions, whereas mitosis only has 1. Meiosis produces those 4 daughter nuclei, and there's 4 daughter cells, where mitosis only has 2 daughter cells. And then in meiosis, remember, those 4 daughter cells are haploid. That means that they have half the number of chromosomes. And in mitosis, they have the same number of chromosomes. And then in meiosis, the daughter cells are genetically not similar to each other, whereas in mitosis, they're genetically identical to each other. So this is kind of a visual breakdown of mitosis versus meiosis. And you can see that there's that splitting into a second set there. So the only real difference between meiosis 1 compared to mitosis is that crossing over happens, where the genetic variability occurs. And the sister chromatids don't separate during anaphase 1. So the genetic material is split in half, but the chromosomes don't pull apart like they do in mitosis. And then meiosis 2 compared to mitosis, the events of meiosis 2 are just like those of mitosis, except for they have a haploid number of chromosomes instead of a diploid number. Mitosis and meiosis occur at different times during the life cycle of the human. It only occurs at certain times. Like I said, for women, you're born with all the eggs that you're going to have. But for men, that spermatogenesis continues to occur. Mitosis is the much more common cellular reproduction. It occurs in all tissues at all times. So you're constantly making new cells. So there has to be some kind of control system for this cell cycle to happen. Otherwise, your cells would just be dividing all the time. And this makes sure that it doesn't happen out of order, it doesn't happen uncontrollably, and that your body maintains the right number of cells. So it has many checkpoints, but there's 3 major checkpoints that we're going to talk about, and that's the G1 checkpoint, the G2 checkpoint, and the mitotic stage checkpoint. So in that G1 phase, remember that's where it's starting to grow and double its organelles. The cell has to commit to divide after this point. If it decides that it doesn't need to divide, like there's enough cells in your hands, then it goes into that G0 stage. It has to have a proper growth signal, usually through some sort of hormone, to be able to pass into that S phase. The second thing that happens is that it checks the integrity of that DNA. So it checks to make sure that that DNA is not damaged. And if it is, then it tries to repair it. And if it's not, then what we call apoptosis or cell death happens. So it kills off that cell because it's no longer, if it split and that DNA wasn't perfect, then it's going to cause some sort of mutation that's going to cause a disease. So it kills it off before it can do that. Then you have the G2 checkpoint. It verifies that it replicated the DNA, and if there's any DNA damage, then it repairs it. And if not, then it does the apoptosis. And then the mitotic stage checkpoint, that's between metaphase and anaphase, and it checks to make sure that all the chromosomes are attached to a spindle. Because if it's not, then what you're going to have is there's going to be extra chromosomes in this cell and less chromosomes in this cell. So it has to check to make sure that all those spindles are attached to those chromosomes so that they'll pull apart perfectly. So there's where your different checkpoints are in the cell cycle. So there's some internal and some external signals. Signals are usually molecules, whether they be proteins or steroids, lipids, carbohydrates. There's some sort of molecule that's going to either stimulate or inhibit that event. So external signals obviously come from outside the cell, and internal signals come from inside the cell. So the kinases are one form of signal, and the cyclins are a different set of signals, and they're an internal signal that are only present in the cell during certain stages of the cell cycle. And destruction of cyclin at the appropriate time is necessary for the normal cell cycle progression. So if that cyclin continues to be present in the cell, then it can't go forward with its mitosis steps. There are some external signals like epidermal growth factor that stimulates your skin to repair itself when there's an injury. Estrogen stimulates the lining of the uterus to divide and prepare for egg implantation. It's what causes your menses. There's contact inhibition. So when cells can touch each other, they stop dividing. And they do that because they realize that there's not enough room for them to continue to grow, and so they stop. So when that happens, apoptosis is what we call programmed cell death. So the remaining cell fragments, so it's going to kill that cell off, and then those remaining cell fragments are eaten or engulfed by white blood cells. The apoptosis happens due to either internal or external signals, and it helps keep the number of cells at an appropriate level. So it's a normal part of growth and development. It can happen to regular cells or like specialized cells like tadpole tails. So the apoptosis happens in their tail as they start to grow because they don't need that tail anymore. So here's your normal cells. In apoptosis, one of those cells needs to die, so the cell starts to round itself and the nucleus collapses. You can see in the beginning that normal cell has that beautiful round nucleus, and then as apoptosis happens, it starts to collapse on itself. And then the chromatin condenses and the DNA fragments. And it's not that you can't reuse those nucleotides, and those pieces of the cell are just recycled to make new cells. So this is what apoptosis looks like, both of the scanning electron microscope and the artist rendering. So what happens if the cell cycle doesn't work? Well, one of the things that can happen is cancer. We've talked about that with the RAS oncogene. If it is turned on, then those cells just continue to produce. They ignore those signals, or those signals don't occur. And so the cells just continue to split and split and split and start to take over the place of normal and good cells. So what happens is there's a mutation, and then that new mutation has the ability to start that tumor. And so you can see it's just a continued, so it takes the place of all of these healthy cells, and it invades and starts to cause damage or take the place of those normal cells. So there's two types of genes that affect cancer. There's proto-oncogenes, and they code for proteins that promote the cell cycle and inhibit apoptosis. And they're like the gas pedal of a car because they accelerate the cell cycle. And then there's tumor suppressor genes, and they code for proteins that inhibit the cell cycles. So when the two are in balance, then your body is in balance. But when those proto-oncogenes mutate, they become cancer-causing genes called oncogenes, just like that RAS oncogene that we looked at in the blast. So when tumor suppressor genes mutate, their products no longer inhibit the cell cycle or promote apoptosis. And so those oncogenes then can just start to produce crazy amounts of cells that cause tumors. Another type of genetic change that can happen is telomere shortening. So telomeres are the very ends of the cells, the chromosomes. And as those shorten, the life of the cell starts to shorten. And that's what happens in normal aging. And if there isn't any of those telomere shortenings, then that can cause some damage to your cells as well. So, characteristics of cancer cells. Carcinogenesis is the development of cancer. That's the scientific name for developing cancer. And the cancer cells lack that differentiation, so they don't contribute to body function. So they don't speak any of the body languages. They don't speak heart. They don't speak liver. They don't speak pancreas. And so they just take up space that could be used by healthy cells. And sometimes they can be immortal, meaning that they don't go through that apoptosis and they just continue to divide and continue to divide. And so here's a picture of what cancer cells look like versus what a normal cell looks like. And you can see that their nucleus looks a little bit weird, and they are starting to take up space where those normal cells normally would be. And so if it's a benign tumor, it's going to be contained within a capsule, which means that at some point they stop because there's no more room. And that capsule is kind of like a hard shell that keeps that cancer confined. Malignant is the bad kind, and it's invasive, and it spreads through all of your tissues and takes up the cell space that could be used by normal healthy cells. So I hope that this helps explain Chapter 5. You're going to have a homework on LearnSmart, and there's also going to be some in-class different activities that we're going to do. If you're on the McAllister campus, there will be some online assignments that I'm going to send via email, so be looking out for those. You guys have a great Labor Day weekend.